Hi everyone, my name is Helena and I'm a second-year applied math PhD student at the University of Washington. Today I will talk about a novel form of biological learning rule inspired from recent experimental evidence. Both experimental and theoretical studies now indicate that classical heavy learning is insufficient, and adaptive learning requires one or more modulatory factors. The additional factors can encode information such as reward prediction error so as to steer the direction and magnitude of change for each individual synaptic weight. Dopamine is considered as a leading candidate for such factor, but there exists many more signals that can be considered as potential factors for learning. Unlike dopamine as top-down learning signals, many are likely to distribute information about local network activity locally in ways that may contribute to learning, which have been neglected in modern biological learning rules. One potential candidate for such local signaling can come from neuropeptides. This is because that, unlike dopamine secreted by designated dopamine neurons, neuropeptide signaling is rather ubiquitous. Recent single-cell RNA-seq findings indicate that nearly all mouse neocortical neurons express at least one neuropeptide precursor and receptor gene. How neuropeptides works is that they're secreted by source neurons and then received by neurons that express the cognate receptor within vicinity. And that can lead to downstream actions similar to dopamine selective GPCRs. So similar to dopamine, neuropeptides are also implicated in plasticity. Moreover, the combinations of neuropeptide precursors and receptors are highly cell type specific. And this creates a modulatory network conveying local network information with connectivity defined by cell type specific receptor affinities. These findings suggest a multi-digraph view of cortical computation and plasticity through a stack of fast synaptic and slow modulatory networks. Motivated by these findings, we test the plausibility of this multi-digraph view by casting these local cell type specific modulatory signals into an explicit learning rule and develop experimentally testable predictions. In particular, we train recurrent spiking network while obeying Dale's law for temporal credit assignment tasks. For clarification, temporal credit assignment deals with determining how much each individual synaptic weight is responsible for the overall network performance so as to guide their individual strengthening and weakening. To solve the temporal credit assignment problem, classical gradient descent learning methods for training RNNs compute the full gradient of weights. And this involves expensive and inaccessible non-local information due to dependencies arising from recurrent connections. The current state-of-art biologically plausible learning rules EPROP is a severe truncation, which restricts the update to depend only on pre- and postsynaptic factors, as well as a top-down instructive signal. Since this learning rule omits a lot of information in the exact gradient, it is no surprise that it significantly underperforms in comparison. Instead, we propose multi-digraph learning, or MDGL, which incorporates diffusive communications through a modulatory network for better approximation of the exact gradient. We note that MDGL can be implemented online and it shares the same complexity as EPROP. The signaling is type specific in the sense that signaling gain value is the same within each cell group. With our addition, classical Hebbian component is not only compounded with a top-down instructive signal as conceived by many modern learning rules, but also integrated with additional factors from cell type specific local modulatory signals. We apply these learning rules to train recurrent spiking network obeying Dale's law. By looking at the learning curves, we observe that the addition of cell type specific local modulatory signals in MDGL resulted in faster reduction loss reduction over training iterations compared to EPROP, the existing biologically plausible learning rule. Repeat this for various other temporal credit assignment tasks spanning seconds and observe similar trends. Thank you very much for listening. Further details of this work are included in additional slides, and happy to discuss those at the meeting. Here is our bioarchive preprint if you're interested.